I will have to wait for the pointer. Doesn't look like highlighter. I'm looking for okay, I will do it like this. Can you see this? My
yes, we can.
cursor? Okay, so you see my screen and my cursor, so then I can give the class. Okay, I'm ready.
Okay. Then allow me to introduce you and then you can start.
Okay.
So Professor De Bazer was trained as an MD and clinical virologist at KU Leuven, Belgium, and did his post-graduation studies from Stanford University Hospital. He co-authored more than 420 publications with 14,654 citations and an H-index of 68. He acted as a mentor of more than 54 PhDs, from whom out of 13 got faculty positions worldwide. He is now leading a research group with a major focus on target identification and validation for diseases with a high unmet medical needs such as HIV, leukemia, and Rett syndrome. He uses both basic and translational research. His group is known for pioneering research in LegF as a cofactor of HIV integrase. He discovered Legend as first-in-class inhibitors of integrase and was the first to propose a functional block and lock HIV cure using Legends. We welcome you, Professor. You
Okay.
can ask.
Yeah, thank you for the nice introduction. So I think we have like 2 hours, so I'll talk on 1 hour. I'll give like a 5-minute break, and then I'll give another 30 minutes, and then I think we have time for questions. I don't know, is it the purpose that people interrupt or they ask the questions at the end?
Yes, I think this is perfect.
Yeah, okay. So I'm giving you a class on HIV. So in the end, I'm going to talk about HIV cured, which is research I'm doing now. But before doing that, because I think the audience may be a bit mixed, I will give you a bit of a historical perspective on HIV, HIV research, HIV treatment. And why doesn't it? Okay, so this is my laser pointer. Yeah, okay. Meeting is being recorded. Okay. And this thing, yeah. Okay, so HIV, I guess it's a familiar face, many people know, but I think we have still many unsolved questions about. These are the numbers of 23. We have about 40 million people that live with HIV. And although we have a treatment, I will talk about, it comes with a high cost of care. Many people die. There was a discussion about how long can people live with HIV. So there's still an issue. Basically, it's a disease where a virus, HIV, infects the immune system, and the immune system actually collapses, and that gives you AIDS. But over the years, 40 years of research, we developed a chronic treatment with antivirals, which is now the standard of care. But of course, the challenge now is to try to find a cure so that we can stop having to give pills to the people. Why doesn't it move? Yeah. Okay, so the overview of the talk, I will give you a bit of a historical perspective. I started to work on HIV as a medical student in '86, and HIV has been identified in 1983. So I have been...
Professor?
Yes?
I think your slide has not moved here. We can still see the previous slides.
Why is this?
I have no idea.
Your screen sharing is paused. Why is this paused?
I think if you start it again, then it will be fine.
Now we see it again?
Wait. Yes. Yes, perfect.
Yeah, but why doesn't it move? Does it move?
Yes, now.
Okay. Anyway, so I will give you an historical perspective since the beginning. I'll talk about the pathogenesis, the treatment, and then I will talk about our own research, targets of the future, LEGEF, and how we discovered LEGEF, how we developed legends, antivirals, and how we go for a cure. So how did it start? 81. A long time ago. Many of you were not born, I think, at that time. Is in California, where some young homosexual gay men were diagnosed with opportunistic infections. These are infections you don't normally see in people with a normal immune system. But here we saw like a pneumocystis carini, we saw candida, we saw CMV infections in normally healthy young men. And some of these people died. So it was a new disease where people got an immune deficiency, and it took a while to find out that this actually was a novel virus. And so the first cases were typically people with this Kaposi sarcoma, which is now we know that it's induced by a herpes virus, virus, sarcoma virus, and is inducing this angiogenesis and this tumors on the skin. Of course, it gave a lot of stigma because people with this kind of sarcoma were known to have this new novel disease. Again, the Kaposi sarcoma on the left, and then this pneumocystis carini, which is a parasite which lives in the lung, severe lung disease. And young people died from it. In Africa, at the same time, the virus was not identified, but many people died. They called it slim disease because people got a lot of tuberculosis, gastrointestinal infections, diarrhea, and they died from an unknown cause. Yeah. As a result, many orphans, because the adults, the parents actually died from the disease, and there were a lot of orphans. So this was the 80s. And then in 83, there was a discovery of the virus. It was a kind of co-discovery. The first one was Luc Montagnier, who is at the bottom, at the top left. And he showed like a figure from his science paper where you see the viruses. It's an electron microscopic picture, and you see the viruses that erupt from the cell. Yeah. And he identified the virus. He actually called it the lymphadenopathy virus. Later it was called the HIV virus. At the same time, it was Robert Gallo. He was in the States at the NIH. 6 months later, he also published the same virus. He called it the HTLV3 virus. In the end, it turned out to be the same virus, and it was called HIV, which stands for the human immunodeficiency virus. Okay, the virus is a... and then, Montagnier and Barré-Sinoussi, they got the Nobel Prize in 2008 for the discovery of HIV. So the virus is a typical retrovirus, and if you know virology, these are enveloped viruses, so they have a phospholipid double membrane. They have envelope proteins, the glycoproteins, which serve to attach the virus to the cell. Inside there is a cone, and inside the cone the viral enzymes, reverse transcriptase, protease, integrase are present, and also the genome, which is an RNA genome. So what is the origin of HIV? I mean, a lot of research has been done, and there is a very related virus, which is the SIV CPZ of the HIV CPZ, chimpanzee. So in the chimpanzee there is a close cousin of HIV. And so the idea is that the virus has jumped from the chimpanzee, probably with a hunter. People eat bushmeat, and they go into the forest and they kill the chimpanzees, and the chimpanzee can bite. And so this way, people got infected. These kind of infections, they happen if people encounter wild animals, but rarely give rise to a pandemic. In that case, early 1900s, I think the socio-economic configuration was present that allowed this to happen. And this was actually the Belgians. The Belgians had a colony, which was Congo. First, it was our king, Leopold II, and later on it was Belgium. And we had the first big city in Africa, which was Leopoldville. Now it's called Kinshasa, and the big port, Matadi. And so they shipped a lot of the rubber and the ivory out of the colony. And so there was a lot of jobs in the city. So a lot of men came out of the villages from the forest, and they lived in Kinshasa. And so probably some of these people got infected with the chimpanzee, brought it to the city. And there, there was also prostitutes from Haiti, that were imported, brought into the city for these men. And these got infected. They infected the men. The men in the holidays went back to their villages and infected their spouses. And there, so in the early 1900s, the virus must have been spreading. And this is the early hidden pandemic. So it took 80 years until the 80s before it was actually realized that this virus was spreading. By that time, the virus has spread in Africa. I told you, the slim disease. But these prostitutes, they were from Haiti. They went back to Haiti, infections in the Caribbean. And from the Caribbean, it went to the States. And it was in the States, like in California, that eventually the virus popped up. People died. And it was realized that the new virus was actually spread. By that time, the virus had spread all over the world and is a pandemic. And many, many people actually have died. And you know that then they make this little tapestry, this kilts for each individual person that dies. So the numbers, this is a bit of an older slide, 2017. I think we have about 40 million people worldwide that live with HIV. And if you see, it's spread all over the world, but most people live in Africa and then mainly in South Africa. This is a picture of a graveyard. So the maximum number of people that were dying was like in the 2009, 2010. In this period in KwaZulu-Natal, South Africa, many people died every year at some time. Young women that went to the consultancy for the babies when they were pregnant, 1 in 2 of them actually were pregnant. And there was no tests being done, so young adolescent people living there, they have the girlfriend. I mean, you had like or a boyfriend, you had like a 50% chance that you were going to be infected. I mean, so the high incidence in young people was so high. Terrible. And so it had a result on the life expectancy. So this is the life expectancy. I think in more developed world, I mean, we live like 60, 70, and now in Belgium, we live about 80 years old. And then you see that there was an increase in this life expectancy also in Africa, but due to the one single virus, I mean, this dropped a lot. I mean, it's increasing again thanks to the treatment. So how can you get HIV? It's mainly through sexual contact, where heterosexual contact, I mean, it has a risk, 1 in 1,000. Homosexual contact has a higher risk. Vertical transmission, which means that a mother who is HIV infected, she can give the virus to the baby during the delivery, also during breastfeeding. And then blood exposure, these are needle stick incidents like doctors and nurses, and then drug use. A lot of people also like in Eastern Europe get HIV by sharing infected needles. Okay, so then for the pathogenesis, replication cycle, and treatment. So the HIV virus is a typical retrovirus, already explained. So it has this envelope, it has this cone, inside the cone 2 RNA molecules. You have the enzymes, the reverse transcriptase is shown, but you also have the integrase, you have the protease, and then you have in the envelope the glycoproteins, which are used to get the virus attached to the cell. The cone is a typical picture on an electron microscopic picture, and a mature virus will have this cone. Immature viruses don't have this typical cone. So the genome is quite known. Like for all retroviruses, it has this GAC, POL, and ENV. GAC are the structural proteins, POL the enzymes, a protease, a reverse transcriptase, and an integrase. And then is the envelope. You have the surface protein and the transmembrane protein, and it's the other structural protein. So it has the long terminal repeats, which are promoter regions, 2 repeats at both ends. And then typical for more complex retroviruses like HIV, it has this additional small genes that encode TAT and REF, necessary for transcription and RNA exports. And then these 4 little genes, NEF, VPR, VPU. It took a long time to investigate and to find out what these genes are doing. So they encode small proteins that actually help the virus to defend itself against host defense mechanisms. I won't go into detail in this talk, but for instance, there is ApoBec, which is a deaminase, which actually can degrade the incoming RNA of the virus. So it's actually the cell protects itself against the virus. But the virus evolved to encode and to produce VIF, and VIF will actually bind to the ApoBec and put the ApoBec in the proteasome so that it's degraded. So it protects actually the virus against host defense mechanisms. And so that makes that HIV is one of it's a recent virus, or a virus that recently jumped from primates to human. So it has definitely evolved to be a very complex, a very adapted, I call it a very clever virus. Of course, viruses cannot be clever, but I mean, I call it the queen of the viruses. It's so specialized to infect, cells of the immune system, which are really very clever, of course, and then to stay permanent in these cells, but I will talk about it later in my class. So it's a really it's a respectful enemy, I would call it. Okay, so the pathogenesis, which is important to understand some of the specificities of HIV, actually you have 2 kind of strains of HIV. And that's because if HIV infects a cell, it uses a host receptor, which is the CD4 receptor, which is typically present on the CD4, which is the helper lymphocyte. But it also needs a co-receptor. And a co-receptor is a chemokine co-receptor. So a receptor which is on the CD4 cell and actually binds chemokines. And you have 2 kinds. You have the CXCR4, and that is present mainly on T lymphocytes, like the CD4 cell. And you have the CCR5, which is mainly present on antigen-presenting cells, macrophages, monocytes, dendritic cells. And an HIV virus that infects a CCR5 cell, we call it an R5 virus. The other virus is the X4 virus. So how do you get infected by sexual transmission, which is the most common route of infection? Through the mucosa, that can be the rectal mucosa with homosexual infection, or heterosexual through the vagina. Then you have these dendritic cells, and the dendritic cell is like a sentinel. It's a cell which protects the mucosa and sees if there is kind of viruses or bacteria. And then this dendritic cell with all these kind of tentacles, it will bind. It normally eats the virus, it destroys it, and it brings it to the lymph node where an antigen response, an immune response, can actually be mounted. So it brings it to the lymph node to have an immune response. Only in the case of HIV, it doesn't work very well, because the HIV just binds the dendritic cell, the DC sign protein, and is actually imported. So here, the dendritic cell is like a Trojan horse, and it will bring the virus to the lymph node where the virus, you here see this dendritic cell, it's covered by viruses, and it just doesn't destroy the virus, but it brings the virus to the place where all these target cells, immune cells, are, and here the infection actually takes place. And from the lymph node, in 2 weeks, the virus will spread all over the body, through the lymph node, in the blood. It goes to the brain, it goes to whatever place, all the lymph nodes, it goes to the bone marrow, and you're infected. But why do I tell this? Because because of this mechanism, it means that through a sexual transmission, you always will be infected by an R5 strain, never by an X4 strain. So the CCR5 receptor, which is present on antigen-presenting cells, 1 or 2% of the Caucasian people, the white people, people living in Western Europe, they have a deletion in the CCR5. And that means it doesn't matter for your normal life, but you're resistant to HIV infection if you have a homozygous deletion in the CCR5. So there are people that have this deletion and that are resistant to HIV infection. It will come back if I talk about the first few patients. Yeah. Of course, does it mean that you cannot be infected with HIV if you have this deletion? That's a question I always ask to my students, so it's less interactive here. But let's say, yes, you can be infected, for instance, if you do intravenous drug use, because then you have blood contact, and their X4 strains can also infect your body. So it's only the sexual transmission which is R5 strains. If you go for intravenous drug use, then you can also be infected by X4. So there are some people that have this deletion. They're naturally resistant to HIV, but still they got infected because they were using drugs, which is not clever. It's never clever, but in this case, it's really stupid to do. Okay, so this is the pathogenesis. And then this is, I think, the most important slide of the introduction on the pathogenesis, because it gives all the features on HIV. So it goes in time, weeks, years, and then months. This is the acute infection. In green, we see the viremia. So we can measure with the PCR, quantitative RT-PCR, the viral load, how much virus in the blood. And then we see that in the acute infection, there's a lot of virus, and then it drops because the immune system will react. We will respond to the virus with our CD4 cells. And the CD4 cell count goes down because many of the cells are killed by the virus. So the virus infects the CD4 cells. Many cells are killed, but you can actually counteract. The bone marrow produces a lot of CD4 cells so that the CD4 count over years more or less stays stable. There is an immune activation, which is in the red, which means that you will produce T cells and also antibodies against the virus. The antibodies will be in the blood 3 weeks after infection, and they are used, as you know, for the diagnosis of HIV infection. So we don't look at the virus in a patient, we look at the antibodies against the virus. And if you have antibodies, you're seropositive, it means you're infected. So during the course, we see that there is an immune activation, and that stays during your life. The viremia goes up and then it goes down, and you have a set point which differs from patient to patient. And the CD4 cell count goes up. So in the beginning of the HIV infection, when we didn't have these PCR tests, we actually thought that the virus was sleeping for years during this chronic infection. It was only after developing viral load testing that we saw that during this chronic phase, there is a lot of virus in the blood, and the virus is actually killing the cells continuously. Only the bone marrow can produce a lot of CD4 cells so that we can keep a steady CD4 cell count. And that's important because that's our immune system. And so what happens is, after years, it can be 5 years, it can be 10 years, it can be 20 years, we see that something is happening. The bone marrow gets exhausted, and we cannot produce enough CD4 cells. It means that your immune system goes down, so the virus goes up. And then you get a steady drop in the CD4 cells, and since the CD4 cell is your immune system, you cannot defend against opportunistic infections, Kaposi sarcoma, Candida, cytomegalovirus, cancers, and you die from AIDS because you're immune deficient. So you do not exactly die from HIV, you die from an opportunistic infection you get because you don't have an immune system anymore. So that's the normal progression of HIV. You see pictures of famous people. This is Freddie Mercury that died from HIV infection. So the diagnosis is based on testing for antibodies. 2 tests in ELISA. which will test if you have antibodies to the P24 antigen. It's a very sensitive, but not so specific. So you can have false positives. It means that if you have a positive ELISA, we always do a second confirmation test, which is a immunoblot or Western blot, so where we separate the viral proteins and then we check for antibodies to different proteins. So it is a much more specific test. And so if a patient has a positive ELISA, and the reference center will do the confirmation test, and it's also positive, then the person is called seropositive, a person that lives with HIV. And then the follow-up we will do the viral load. We will see how much virus is in the blood. We will test how many CD4 cells are left, because it means the immune system, and we can do some more specialized tests, but I won't talk about that. Okay, so if we now know that this is the natural progression of HIV, acute infection, which can actually be associated with some symptoms, but atypical symptoms, rash, fever, lymph node swelling, like a flu-like, it's not specific, but not some people really asymptomatic. Then a chronic phase, no symptoms, you don't know if you're not tested that you have HIV, and then there is AIDS. So then the question is, how can we treat HIV? So this is actually the comparison of the status of HIV during these chronic years. So it's a bath tube, where the water in the bath is actually your immune system, is how many CD4 cells do you have. And if you have enough, you have an immune system. You don't have, if you go below, you get AIDS. So we have to see that the level is high enough. And this is, on the one hand, you lose CD4 cells because the virus is killing the cells. So So the bath tube is emptied continuously, but the tap is open, so you always make new CD4 cells from your bone marrow. And so making new cells and losing cells is this equilibrium, and the status is different from patient to patient, but it has to be low, above a threshold, otherwise you get AIDS. And so if you know this, you can think theoretical about two solutions for HIV. One would be, let's open the tap. Let's make more CD4 cells. And we have tried by giving, for instance, interleukin-2, stimulating the bone marrow, but bad idea. Because you actually feed the virus, you give them more cells so they can evenly grow more rapid. So it's not a good idea. The only good idea is to stop the drain, stop the replication of the virus so that the cells are not killed any longer. And how do we stop the virus? With antivirals, specific molecules that block the virus replication. Okay, and that's what we did. So from the 80s on, we first developed AZT, which is a reverse transcriptase inhibitor. But when we gave this to the patients and we measured the viral load, we saw that the viral load went down, but after 1 or 2 months went up again. It didn't work. Then we developed protease inhibitors. Then we developed non-nucleoside RT inhibitors. And then we said, and David Ho was the guy, let's combine 3 drugs at least. And why do we combine? Why do we do a combination treatment? It's because of the resistance of the virus. Because HIV has a high replication rate, but also has a low replication fidelity. It means that the reverse transcriptase makes many mistakes. You maybe know from other classes that the DNA polymerases in the cell, which copy your DNA, they have a very high fidelity. They don't make mistakes. They cannot make mistakes. You don't want to get mutations in your cells. You would get cancer or a disease. Because the DNA polymerase has a proofreading capacity. If it incorporates a base, it will check if it's the correct one. If it's not the correct one, it will cut it out. Reverse transcriptase cannot do that. It has evolved to make mistakes. And why does it make mistakes? Because it changes the genome, and so it can change its envelope. And so by changing the envelope, it can escape the immune system. So it's very good for the virus to do that. And we can calculate that so that the reverse transcriptase will make a mutation on every 10 to the fourth bases. So we know that the genome is 10 kb. It means that a genome of HIV which is copied will contain one error. We know that we make 1 billion virions every day. So we can calculate that every site in the genome per day has 10 different mutations. So it means if you're infected with HIV, you're not infected with one strain, you're infected with a swarm of all little different viruses. And so when there is a drug coming in, you will have a virus which has natural resistance to this drug. And so it means that all the other viruses, they cannot replicate, but the one which is resistant, yeah, will be selected. So, we can calculate in 2 weeks to 4 weeks, this resistant virus will take over all the other viruses. So this is evolution on the lab scale in the patient. This is evolution on a weekly base. So the evolution, the theory of Darwin, that there is mutation and then the selection and the survival of the fittest. This is happening in HIV. So it has this intrinsic making of mistakes, and the mistakes that lead to a virus which is resistant, that will be selected and that will win. Okay? So if we know that, we say, let's do a combination treatment. Let's give 3 drugs, because then the virus has to be resistant to 3 different drugs. So it has to have mutations on 3 different spots in the genome, which is very unlikely. Plus, if you give 3 drugs, it will be a very strong suppression of the viral replication. If there is no viral replication, there is no reverse transcription, mistakes cannot be made. Yeah? So this is the double reason why we do the combination treatment. And this was successful. By combining 3 drugs, we can reduce the viral load to undetectable levels. So we don't detect it anymore in the blood. Actually, people have to take the drugs, but people that have a stable relation, they can even have intercourse, people can have a baby, and there will be no infection because the virus is so low. Yeah, so this is really a big success of this antiviral treatment. It was so successful that it's also used for other viruses. Maybe you know, like hepatitis C virus. We also give a combination treatment, and there we can even eradicate the hepatitis C virus after a course of 3 months, because the virus is not integrating in the genome, so we can actually destroy all the virus. But the combination treatment is important, and this is David Ho in 1996. He was the man of the year in Time because he was the one who advocated actually to use these cocktails of drugs. So which drugs have been used over the years? Again, I go quite superficially. This is a replication cycle of the virus. The virus, as I told, will interact with the GP120, with the CD4. It will also interact with the co-receptor, CXCR4 or CCR5. And then the virus will enter the cytoplasm, where the RNA of the virus is copied into a double-stranded DNA by the reverse transcriptase. The virus will be imported in the cell, active nuclear import, and it will be integrated in the cellular DNA. Once it's integrated, it's considered a gene, so it will be transcribed. You have mRNA, genomic RNA. There will be proteins being made that actually sit under the membrane of the cell, and then the virus will be producing new viral particles, and there the protease will actually process the polyproteins into mature proteins, and then you get this nice cone in the virus. And so over the years, we developed drugs mainly targeting the viral enzymes. So we started with the reverse transcriptase inhibitors, and we had nucleosides that block the chain termination, the reverse transcription. Then we had the non-nucleosides. Myself, in my PhD, developed the first non-nucleoside RT inhibitor. And then we had the protease inhibitors that work at this late stage. And then in the 2000s, we had the integrase inhibitors, and I'm also working on integrase inhibitors. And now they are the best drugs, or actually these integrase inhibitors. Very recently, it's still not on the picture, we also developed capsid inhibitors that actually block the entry of the virus, lenacapavir from Gilead, and that's actually now the best drug in the market. Okay, fusion inhibitors have been developed, like Maraviroc. The problem was it's only working on R5 strains, so you have to actually type the virus before you can actually use the drug. And so we have over the years developed many drugs, single pills, combination pills. I won't into detail. In the beginning, people have to take, because you need 3 drugs, some of them 4 times a day, sometimes with the food, with the meal, without the meal. So very complicated regimens, but pharma companies have done a really great job. They combined these drugs into one pill once a day. One pill once a day is now the standard treatment for HIV. And they went even further. So now we have these long-acting drugs, where drugs are given through injection, where you only once in 3 months have to the doctor where you get your shot, and then you get your 3 drugs as a depot, a long-acting drug. Okay, in the beginning, these drugs came with a lot of side effects, but that also this has improved a lot in the new drugs that are available. So what is the aim of the treatment? So now the standard care is to treat all people as soon as they are diagnosed. The idea is that if you treat early, you can reduce the reservoir, and you have a normal immune system, improved life quality, improved life expectancy. Still a bit of a debate, I think, because, I mean, we have not treated people with the best drugs for a whole lifespan yet. I think we are about 20 years now. So we do not know what will be the long-term effects of the treatment and the long-term effects of the chronic inflammation. Because people living with HIV, even if there is no virus in the blood, they have inflammation. And inflammation can lead to premature aging, heart, brain. So there may be some issues. See, the other issues, of course, that people have to take the pill every day. So there is adherence. If you forget to take your pill, then the virus may come up and get resistant. And some of the drugs come with side effects, and also the high cost, of course. So some of these side effects over the years, you had problems with the lipids, mainly like the nucleotides like tenofovir. Lipodystrophy means that the fat distribution changed, very skinny, but then a hump on the back with the protease inhibitors, liver toxicity, kidney problems also with tenofovir, osteoporosis, tenofovir, CNS and NRTIs gave like psychotic problems sometimes, and a lot of drugs came with diarrhea. But also there, companies did a lot of development work on the drugs, and now we have one pill a day with really limited side effects. Okay, so then who can get the drug? So you see that the availability of the drug worldwide has increased a lot, as it is a bit of an old slide. So we call it expanded access. And so there are programs, development programs, there are international foundations that pay for these drugs. So people in the West, they pay the full cost, but people in developing countries, they just get the marginal cost, so it's cheaper for them. And so the aims, we had the millennial goals, was to get 90% of the people that have HIV diagnosed, 90% on treatment, and 90% on below detectable levels. We didn't reach that, but we came close. So it's an enormous, I mean, I think progress in, I mean, if you consider 40 years of HIV research, we went from a new virus to novel treatment, to chronic treatment, and expanded access. So I think it's one formidable exploit that humankind have actually done for human health. Okay, so if we now look at the most recent data, so we have 40 million people living with HIV in 23. We still see that 1 million people are getting HIV, so we still have new infections, so we still do not control the pandemic completely, and we still have about 600,000 people a year that die from HIV. So HIV is still a pandemic. So there's a lot of commotion, a lot of problems, of course, also with the COVID and the corona, but that's over. So now HIV is really the pandemic. And so this is the expanded access, where you see that about 80% of the people with HIV now get combination treatment, is available, and are under treatment, which is a formidable success, but it should go even higher. And so as a result of this formidable exploit of of understanding HIV, developing antivirals, and giving access to the people, we see that the incidence of new infections is dropping. So we have less new infections and less people die. And so the people don't die if they are treated because they can suppress the virus. And then why is there less infections? We call it treatment as prevention. If we can treat the people, they will have less virus and they will be less likely to infect other people. And so treating all the people with HIV will be able to prevent new infections. And that's actually what's going on. So these slopes are going down. But we still have some decades, I think, before we can actually close this. The other issue is that since people don't die from HIV, which is a good thing, these people will live with HIV for the rest of their life. So all the people that have HIV, they will live with HIV for the rest of their life. And the only way to solve that is to find a cure for HIV, to eradicate HIV from these people. And so still, even if we can give access to all people and prevent new transmissions, we still will have millions of people with HIV that or we keep on drugs for the rest of their life, also at a very high cost, or we try to find a cure to definitely cure it. Okay, so then I will come to my third part of the talk, which will be on LEDGF. And so I propose I break here for like 5 minutes, so about half of the talk, and then you can have a toilet break if you want, and then I will be back and we will discuss my own research, which will be on LEDGF, and I will be historical. We identified this protein in 2002 as a host factor of HIV. I will tell you how we identified, how we developed legends, and then how we now use it for cure strategy. So don't run away. Well, you can run away for a short time, but I will be back 10 to 11. Okay, see you. Stay tuned.
Thank you, Professor. See you. Thank you. Yes.
Because I don't see my screen, which is a bit difficult. You can see my screen now?
We can see your screen, but not the PPT. I think you are sharing a different screen.
Is Type share. Wacht, share screen.
Yes. No. Maybe you can minimize your browser.
I don't understand why this has changed. View.
उनको नजर आ रहा है जो वो शेयर कर रहे हैं, शेयर करने वाले को भी नजर आ रहा है। वो ऊपर आएगा साइड से वीडियो आ जाए हम सारे लॉस कर दें बात पाकिस्तानी होते हैं जुगाड़ी होते हैं हाँ
ben in een webinar bezig, Brigitte. So. Now, now you can see it?
Yes, it's fine now.
It's fine?
Yes.
But I cannot see my slides, so that's very difficult to... Uh, why does this... Wait, I will try again.
क्या वो पाकिस्तानी नहीं हमको जुड़ा कर तो नहीं था
This one. No.
We can now see your screen.
Yes, but I can see it only very small. I don't know what happened. It should be... Why is it so small? Okay, I will try like this. Yeah, and it doesn't move. Why is this?
You have stopped sharing.
Yeah, now I stopped, but I tried to get it, but for some reason, slide I get is very, very small.
But here it is fine.
Yes, but I cannot see my slides, so it's difficult to talk. And I cannot move the slides either. They don't move.
Maybe you can connect again.
Yeah, I will go out and get again. There is something wrong with the connection. I don't understand.
Maybe you can connect again and it will be better. We are now restarting the meeting, so we will join you in a while. हाँ मुझे पता है यहाँ समझता नहीं है लेकिन फिर
I I entered through a different site, but okay. Good. Thanks for holding on. Part two will be about our own research on LEDGF as an HIV cofactor. Again, this is the HIV replication cycle, as I told. So the virus enters, then the RNA will be transcribed by the reverse transcriptase and then imported into the nucleus where the virus is inserted, the DNA. Once the virus is in the nucleus, it's considered as a gene. There will be normal transcription, production of viral proteins. My group, since end 90s, has been interested in the integration step, which is the step where the virus will bind the full-length DNA. We believe that the integrase is involved also in the import of the virus into the nucleus and then the insertion of the DNA in the cellular DNA. And so we, from the beginning, had the idea that this integration step, which is quite complicated, is not only done by the integrase, but also is using some cellular proteins, which we call host factors. And so we have been actively looking for cellular proteins that assist the integration step. And so the one we found was LEDGF. So LEDGF was found by a co-immunoprecipitation. So we produced cells where the integrase is encoded by a synthetic gene. And the integrase is produced with a flag tag. So we can actually make a lysate of the cells and then use antibodies to the flag and precipitate integrase to the bottom. And then we take the precipitate and put it on an SDS page. And then you see the light and the heavy chain of the antibody. We see the integrase, and then we see other proteins that were bound to the integrase, which we call binding partners. And we had a band of 76 kilodalton. We cut it out. We did N-terminal sequencing. We did mass spec. And so we identified this protein as LEDGF. So it was a protein known. It was not a novel protein, but it was the first time that it was linked to HIV. And so when we look at the location in the nucleus of the cell, we see that the integrase is located in the nucleus, and with antibodies to LEDGF, we see that also LEDGF is located in the nucleus, and they actually co-stain. So they're sitting together in the nucleus. So again, they are close binding partners. And then, of course, if you know that the protein is sticking to your integrase, it's a binding partner, but you do not know if it's a real host factor. For a host factor, you need something functional. And so we did this mini HIV DNA assay. It's an agarose gel. So this is DNA, which has the LTRs of HIV, but it's shorter. It's a half-side genome. And if we add integrase, then this HIV will be inserted into each other. And you get these strand transfer products that you see here. Now, if we add LEDGF, recombinant LEDGF, we see that you get an increase in the strand transfer products. So it means that the protein not only binds the integrase, but it also will stimulate integration, meaning it's a host factor. And so we called it LEDGF. So the name stands for the lens epithelium derived growth factor. So it was an ophthalmologist who identified the protein in the epithelium of the lens. It's not such a good name because the protein is present in all the cells, so it's not specific for the lens epithelium, and it's not a growth factor. But we keep it under its initial acronym, LEDGF, because it's a unique acronym. If you go to Google or keywords or PubMed, you will find this protein. And so initially it was described as a cellular stress response protein in cells, so without HIV infection, and a transcriptional coactivator. So the protein comes in two flavors, a long version and a short version, P75 and P52. In the C-terminus of the long version, there is this protein binding domain. We call it an integrase binding domain, or IBD, which is binding the integrase. In the N-terminus, there is this PWWP domain, and this stands for the amino acids proline and tryptophan. That makes a cage, an aromatic cage, which interacts with histone marks. You know epigenetics, which is the methylation of acetylation of histones, and that regulates transcription. Well, one specific mark, H3, dus on the third histone, on the 36th lysine, if it's triple methylated, this is a very specific mark for active transcription. And so this PWP is recognizing this. And so what LEGEF is actually doing is it's on the one hand, with the tail, the C terminus, it's binding the integrase, and with the N terminus, it's recognizing active transcription. So some nucleosome histone features that tell you this part of the genome is active transcription. And so by doing that, LEGEF will bring the integrase to these sites. And so HIV can integrate in this active transcription, which is a really clever, again, way of evolution, because by doing that, HIV knows that it will be an active DNA, and so there is transcription, and there will be copies of the virus being made. I mean, the virus will make babies, let's say, because it's integrating in an active part of the genome. And so LEGEF is a tether. It brings the HIV to the genome, but it's also like a GPS. It will tell you where the best sites in the genome are. Yeah? And so the virus evolved to use this to find the best places in the genome. And so the P52 is not a cofactor. So lots of research in the early 2000s to prove and to validate that LEGEF is a real cofactor. Finally, my former postdoc, Peter Cherepanov, he solved a co-crystal of the core domain of the integrase. So this is the integrase, has an N terminus, it has a C terminus, it has a catalytic core domain, which is doing the catalysis of the integration reaction, yeah, with the DDE motif. And then it has this IBD, the protein binding domain, which is a 4 alpha helical bundle with a loop which is protruding in a pocket at the dimeric interface. Yeah, so this is the interaction. If you see this is a protein-protein interaction and you tell this to a modeler, he will get excited because this is a very defined molecular interaction. It's not a flat surface. It's a very defined interaction that if you can design a small molecule to fit in the pocket, you will be able to overcome the binding energy and actually develop a drug. And that's what we eventually did. Okay? Now, before starting the drug discovery, we needed to validate that LEGEF is in fact a target for HIV. And we did this by this experiment. We actually overexpressed the tail of LEGEF, the IBD or the delta 325, in cells, and then see what happens with HIV. So in wild-type cells, you have HIV infection. We measure P24. If you express GFP, you have HIV infection. But if you express delta 325, this, or IBD, this, you have a block in HIV replication. This is a logarithmic scale. And then we had one specific mutant, which is here at the top of the loop, the position 366. Yeah? We put aspartic acid into an alanine, and then we know that you lose the interaction with the pocket, and you lose also the inhibition. So this is a very, very fine control that this is a specific interaction and a specific inhibition. And the cartoon explains you even better the how it works. So normally the LEGEF will bind with the head to the chromatin, yeah, to the nucleosomes, and with the tail to the integrase. But if you express only the tail will bind to the integrase, but cannot bind to the chromatin, so it will block the interaction of HIV and it will block HIV replication. So in doing that, we validated LEGEF as an antiviral target, but we also proved the mechanism. HIV, LEGEF is a tether. It brings the ship of the integration, yeah, to the chromatin, which is actually the harbor. And so the rope is actually what this LEGEF is doing. Okay, so we validated LEGEF as a target. We had a co-crystal structure, and then we went into the development of legends as antivirals. So there was a PhD student who designed a pharmacophore. He said, let's make an ideal inhibitor that sit in the pocket of the integrase, and it has these features of aromatic hydrogen bridges, large, small, whatever. And he made a virtual molecule, and then he looked in the database of structures that were made mainly in Russia at that time. Yeah? And he did a virtual screen. So he said, like, my compound, is there anything similar in the database? And he found 25 compounds that he ordered and we tested. And one of these compounds had a very low activity. It could block the interaction between integrase and LegF at 100 micromolar, 36%. So you really have to believe in your project to continue with that. But that's what we did. So, and what was actually the screening cycle? So we had this pharmacophore model, which came from structure-based or rational drug design. So we designed the molecule. We had this alpha screen, which is a bead-based assay. You have two beads. One bead you couple LegF, one bead you couple integrase, and you put it in a machine. And if you excite the donor, there will be an oxygen transmission and a light emission that you can measure. If you block the interaction between LegF and integrase, so your light signal will drop. So you can easily screen for inhibitors of the interaction. Once we have an inhibitor, we also test for anti-HIV activity. It's a classical multiple round assay in MT4 cells with a vital dye staining. Yeah? So normally the cells die because of HIV, you get a yellow color. If the cells are alive because of the compound, you get a blue color. And you can also test the toxicity. Yeah? This data, activity, in vitro activity in cell culture, we give to the drug designer who modifies the compound, the chemist, who will make new molecules, and eventually also the crystallographer who crystallizes the compound. And then with higher resolution, we can also make subtle changes to the compound. And so that's what we did initially with this virtual screen. Initial compound, very low activity. And then the chemists, they made some changes. Yeah? So here they changed this phenyl, and then eventually they replaced these side chain by carboxylic acid. And so the carboxylic acid is a typical feature of all these legends. And this was the first compound which showed activity in alpha screen, in vitro, and also very low activity in cell culture. And then we optimized the molecule. So this is all different derivatives. And you see that the IC50, so the concentration you need to inhibit, is going down. And also the selectivity is going up. So they are active against HIV replication. They're not toxic. And so we reach a selectivity index of 25 with this compound. And with this compound, we could then actually do some characterization. So we found that the virus, that the compound is active against strains that are resistant to raltegravir. Raltegravir is the classical integrase inhibitor, which is on the market. So if the virus has this mutation, it loses activity. Like these two typical mutations, this virus is 500-fold less sensitive, but our compound is still active. So no cross-resistance, because our compound binds to a different pocket. And we actually could show that the molecule is sitting in the pocket, and explain you that on the loop of the IBD, which is in gray, there is this aspartic acid. Well, our compound has all an a carboxylic acid, and our compounds mimic this aspartic acid. So they are like mini IBDs, and they compete with the IBD binding to the pocket because of this carboxylic acid. So there is like, they sit into the pocket and the IBD cannot bind, and it actually has a similar carboxylic acid at this position. And so we developed these compounds in 2010. It was a nice paper in Nature Chemical Biology. We also had IP. I mean, we licensed our compound to Pfizer. And then many, we call the compound legends because they block the interaction between LegF and integrase. Also, it's a bit legend in the word, so we hope they're once a legend, legendary compounds. So we call them legends. And many companies developed legends, which come with different names, Alinis, Nikinis, Bayais, they're all legends, and they all have this carboxylic acid, okay? Okay, so then once we had the compounds, we published the compounds, we did actually research with the compounds. And one particular thing if you have a compound, of course, you go to the clinical development, but also you use your compound as a kind of research tool to do research on HIV. And so we found that if you take a cell which is infected with HIV and you add the integrase inhibitor, raltegravir, it has no effect because integration is an early step. Ritonavir is a protease inhibitor. It will affect the production of infectious virus. And what we saw with our legend, that if we add a legend to a productively infected cell, it does not affect the production of the virus, but the virus which is produced is dead. And this was something strange. How can an integrase inhibitor work on the assembly of the virus? And what we did is electron microscopy, and I again explained you that normally these viruses have this nice cone. But what we saw in the presence of the legend is that the cone is outside of the content is outside the cone, or there is no cone. So it seems that by binding to this pocket of LEGEF during the assembly, legends interfere with the normal production of the virus, and you have dead virus. Yeah. So we learned that integrase plays a role during assembly, which we did not know. So we had to develop a drug to identify a mechanism. And secondly, now we have a compound which acts early on integration, but also late on assembly, which makes it a very good antiviral. Good. Then the last part of the presentation is about the functional cure. I promised you to talk about HIV cure. So what is the problem with HIV? So we know that we can treat an HIV infection and that we can suppress HIV. This is the viral load. But HIV inserts itself into the genome, that's one, and in the genome of memory cells, so CD4 cells, immune cells, that actually live as long as the patient lives, because these are the immune memory cells. This is the normal task of these cells, to defend yourself against an enemy. You mount immune response, and then the enemy is gone. You go asleep, but you remember. And when the enemy comes again, you can activate the response. Well, if HIV is again clever to have evolved to infect immune cells, integrate the immune system, stay latent, so the immune system cannot recognize, and do this in long-living memory cells. So if you stop the treatment and your memory cell meets the normal pathogen, the microbe, the bacteria, the virus, then this memory cell will get awake, will replicate, and then the virus will replicate as well. And that's what we see. If we stop the treatment of HIV, then the virus comes up in 2 weeks, 4 weeks, 2 months, and you see a rebound of the virus. So we cannot stop giving the pills to the patients because the virus will come up again. And that's the difference with hepatitis C, where we can eradicate the virus. And so these cells that carry the latent HIV, they are in the blood, they are on the lymph node, they are on different places in the virus. We call this the reservoir. And so this latent reservoir is the reason we cannot cure HIV. And so there is a lot of research going on to identify and to try to cure HIV infection. So different approaches are being done. With small molecules, there are 2 different approaches. One is the purge and kill, and one is the block and lock. So the purge and kill has the idea we add what we call a latency reactivating agent. So we say the virus is asleep. Let's stimulate the virus so that it pops up again. And when it pops up again, it will be toxic for the reservoir cell, or the immune system can kill. So we call it purge and kill. Lots of research, clinical tests, but not so successful in the clinic. So we cannot activate all these viruses, apparently. So we developed another strategy, which we call block and lock. This is with the legends, and the idea is, let's give a treatment so that the virus stays asleep. So that if you take away the drug, the virus comes not up again. So it's the opposite of the shock and kill. Instead of like getting the virus out, we put it in a big sleep. And so what was the idea? So the idea is that we know that LEGEF is the tether of HIV, but also determines where HIV integrates into the genome. It binds the PWP to this H3K6 metal tree. And the IBD interacts with integrase. And we know that if we reduce LEGEF, we get out of transcription units, we can retarget integration. Yeah, so LegF is a kind of GPS. So we know now that we have these legends that are inhibitors of integrase LegF. So the idea is, if we now add legends during HIV infection, we will interfere with the GPS of the virus. What will happen with the virus? Where will it be integrated? And if it's integrated, is it still active or not? And so we sequenced integration sites, complicated slides, heat table, but here you can see normally the virus is in RefSec genes, which means active genes. But if you knock down LegF, it drops. Well, if you add legends, we see a shift. So the virus is not any longer in these active sites. So if you interfere with the GPS, the virus gets lost and it gets in another site. And then, of course, the question is what happens with the virus. For that, we use this double reporter virus. It's a virus that has two reporter proteins, a red fluorescent protein and a green. The red fluorescent protein is encoded by a housekeeping gene. It means if you infect your cells, all cells will have red protein. The green protein is encoded by the LTR. It's only active in active cells. Well, the LTR is active. And so cells that are infected are red, cells that are productively infected, they are red plus green is yellow. Yeah. And so we can separate sleeping cells from active cells. Yeah. And that's what we did. So if we add legend during infection with this OGH reporter virus, we see, of course, legends inhibit integration. You have less virus. Yeah. But if you look at the different proportions, this is an infection without drug. So we have the red fluorescent protein to green, which is the double positive. If you add 50 micromolar legend, you have much less integration. So it's dropped because there is inhibition. But if you look at the proportion, you will see that from the double positive, you go from 5% to 0.03. But for the single positive, you go from 15 to 5. So if you look at the proportion, yeah, the quiescent versus the total, yeah, you see that it goes up. Yeah? It's about 70% of the cells which are asleep. If you add legends, you see that 100% of the cells are asleep. So it moves that if you interfere with the GPS, the virus comes at the wrong side. At this side, it will be not active. It will be asleep. And then the main question is, if we now reactivate the virus, which is mimicking your memory cell that gets activated. So we take cells, we infect them with OGH in the presence of a legend. We take away the legend. We wait 2 weeks. Then the virus will be silent. Yeah, it will be quiescent. And then we reactivate the virus. And then we see that we can, in this case, activate for twofold, 100% of the virus, yeah, twofold more of these productively infected cells. But if we have treated in the presence of a high concentration of legend, it doesn't happen anymore. So the virus, which is at the wrong side, is asleep and it cannot be awake. Yeah? So this is what we call the block and lock phenotype. It's a block because there is less transcription, and it's a lock because we cannot move them out of the sleep. Yeah, and this is what we called a block and lock phenotype. So if you can interfere with this transcription of the virus, then you can put the virus asleep, and that's another way of actually achieving a cure. Yeah, and instead of a shock and kill, we have a block and lock. So we proposed this in 2016. So we have a paper on the block and lock cure strategy, but people were very skeptical about this. They said like, yeah, but it's dangerous. You do not eradicate the virus. The virus will be still there. It's not ethical, and so on and so on. So in the years followed, and now we will go a bit quicker, we investigated the mechanism. Two PhD students, Gerlinde and Julie, they looked at the mechanism. How does it come that a virus which is not directed by LegF is less active? And we measured the integration sites of the different viruses, and we saw indeed that normally the virus is integrating close to the H3K6-methyl-3 because this is the site which is recognized by the PWP. You remember? We see that in the presence of legend, the virus is away from the legend, from the H3K6-methyl-3. Yeah? It's not away from an acetylation marker, but it's away from this specific marker. Yeah? And that means that more of the virus will have no expression. And so we measured this by what we call beehive. It's a technique where we use a barcode in a virus, so we can actually then look at each virus, because it has a barcode, where is the virus? And this is, for instance, chromosome 2, and each band represents an integration site. But since the virus will also have mRNA with the same barcode, and we can determine the RNA level, we can actually link each site with the RNA level. So this one has a high, this is a low, this is a high RNA, and so we can say the RNA level per copy. And then we see that if we add legend, these levels go down. Less integration, because there is inhibition, but also less virus. And at the highest concentration, very few of them, they are like resistant, so they have some residual ones, but most of them are completely silenced. Yeah, and this is also shown in these slides. If you want to know, I think you can get the slides. Okay, so this gave us this kind of cartoon that in the normal default mechanism, LEDGF in yellow will guide HIV to this H3K6-methyl-3 on the nucleosome, which is the active site. Yeah, if you add legend, this is gone, and then the virus will drift. But if the virus happens to integrate close to enhancer sites, enhancer is a site in the genome, it's known from the cancer biology that it has a lot of transcription factors, so it's an active site as well, then you can also have some HIV, and these are these residual ones. So the default is LEGF, and we can treat with legend, and there is a secondary like escape mechanism for the virus. Okay, then Julie, she used a kind of advanced FISH technique to image DNA and RNA in cells. So each red dot is a virus, and the green dots are the RNA molecules. And you see that if you add TNF alpha, you get more green because reactivate the virus. But you see that if you do this experiment in the presence of a legend, there is less transcription and there is less reactivation. Yeah? And we quantify this in this kind of representation. So we show per cell, so each dot represents one infected cell, how many RNA molecules are there in the cell. And you see the control, and if we reactivate, you have more RNA, yeah? Because they are activated. But you see that in the presence of increasing concentration legend, there is less RNA, so we have a block. And if you then reactivate, you have less reactivation. So you have the block and the lock, less RNA in the cell, less reactivation. But you still see some few residual ones, and these we think are these ones that go close to the enhancer. This I will skip. We did the same experiments with a compound from Gilead. You know, it's a big pharma company, and this one is active in nanomolar range, and we show the same effect. So this block and lock can also be achieved at nanomolar concentration. Okay, I told you that people were very skeptical when we proposed this strategy, because we keep the virus, although we put it to sleep, until early 2020, when Matthias Lichterfeld in America, he studies elite controllers. And these are people that can actually control HIV to non-detectable levels without antiviral treatment. And he looked at integration sites in these people, and he saw that over time, yeah, these people get more and more integration in inactive genes, in heterochromatin. Yeah? And so he made this theory of the natural block and lock phenotype, that if in a patient a virus is located in active gene, which is the default pathway, this will give expression, and these cells will be killed by the immune system or by the toxicity. And so you lose these cells, while the virus, which is located at the wrong site, an inactive site, you will keep those because they are inactive. And so it shows that this block and lock is a natural phenomenon. There is a selection going on, only it takes 20, 30 years. Yeah? And so we should facilitate, but it's not dangerous. And so what we do with trying to mimic this block and lock is actually a natural mechanism. So once this paper came out, I mean, our strategy was accepted by the field because it seems to be a natural mechanism which is also safe. And so the final part is a recent paper that we put in Nature Communication. We had a lot of media coverage. And so we did something for this residual expressor. So I told you that the default pathway is integrating close to the nucleosome. But if you happen to integrate close to enhancer, you still will have expression. So enhancers are studied in the context of cancer, and people develop enhancer blockers. So one of these are inhibitors of the BRD4 molecule. Yeah? And you have different kinds. You have JQ1, which binds to the BD1 and the BD2. And then you have this compound, which has been proposed as a latency promoting agent. So it's also a block and lock compound by blocking BRD4. And so we investigate this, and we confirmed that while the JQ1 is actually stimulating transcription of HIV, we see that the ZL050 inhibits HIV. And we show this also with co-localization. We show that if you look at a location, so the BRD4 is typically located at acetylation marker. With JQ1, you get an inhibition. And with ZL, you get a stimulation of the location. So they have a different mechanism. They inhibit TAT-dependent transcription. Yeah? And so then the main experiment was this one, where we showed in primary blood lymphocytes of patients, if we infect with HIV, yeah, we can actually reduce transcription of the HIV. So they also act as block and lock agents. We can add them multiple times. We have a stronger effect. And then if we remove the compound, we still see a residual effect. And then the final experiment, yeah, what happens if we combine the legend, which is blocking integration, with the enhancer blocker? We get an additive effect. We really can block also the residual integration, which was the purpose, so which we call the enhanced block and lock. So with this, I would like to thank you. So these are the people which are, of course, I presented you the data over 20 years of LDGF research. Not all the people are on the picture, but some of the people which are now at present in my lab are in these pictures. And so I would like to thank you, and I'm happy to take any questions.
Thank you so much, Professor. This was really an interesting lecture.
Okay,
And do
thank
you
you.
have any questions? Questions? Questions?
Good morning. Good morning.
Yes.
Okay, so good morning, Professor.
Good morning.
Thank you so much for that beautiful presentation and very interesting results. Mine is like quite simple. I just want to understand if you, by any chance, were able to try and research on the effect of the legends, that the effects that they would have on the normal host activities or the host functions of the lens protein in during like the normal transcription activities of the cells that are host related and not virus related.
Yeah. Good question. So the question is, these legends, do they also have effects on LEGF? Because LEGF, of course, is a cellular protein. I didn't talk about it, but LEGF is in the cell without HIV, and it also brings other proteins to the chromatin. Well, the good news is that legends are sitting into the pocket of the integrase, so they do not bind to the LEGF, so they don't have any effect on LEGF. They're perfectly safe. So they do not bind to LEGF. They sit only in integrase, and they are specific for HIV integrase.
Okay. So, do you know if there is any chance that the HIV virus can develop resistance, like towards that binding?
Yes, okay, very good question. So the reason why we actually developed legends and we looked for host factors was to try to avoid resistance, because a host factor, I mean, does not mutate, because it's a cellular protein. It's replicated by the DNA polymerase, so it is very conserved. And so if it has to bind, so the site of integrase that binds to the host factor is also conserved, because it has to bind to a conserved protein. And so the idea was that if we find interface between a viral protein and a host factor, this would be more difficult to get resistance. So this is one of the theoretical concepts. Still, and you can find it in our paper in Nature Chemical Biology, I think for all antivirals that are true antivirals, it is possible to select resistance in cell culture. And so we did. It's not easy. You need multiple passages of the virus in the presence of the drug, but we find some mutations like the A128T mutation arises, and it can give resistance to the legends. So there are some natural polymorphism in the pocket in patients. People have looked at that, but they have developed legends that actually can overcome these polymorphisms. But I think it's possible to get resistance. I mean, we do not know from the clinic yet, but anyway, they would be given a combination treatment to avoid resistance. Yeah.
And has there been any work to test the performance with the proposed combination drugs?
Yes, we have done in our paper in 2012, we did combination in cell culture, and we see there is no antagonistic effect, so there is additive effect of compounds with protease inhibitors, nucleosides, and so on. Yeah.
Okay.
They can be combined. Yeah.
Thank you. Are there any more? Yes, there's one more question.
Thank you, Professor, for your lecture. I want to ask, I could understand that the study, there was a molecular docking that was done to identify the drugs. And after the drugs were identified, there was also molecular docking to ascertain the effectiveness. So what was actually the reason for that? Was it predative, or was it predicted that the compound will not be active?
It's not, your question was not completely right. So it's correct that we did a virtual screen. So we designed a molecule and we used docking to identify a pharmacophore, and then to screen virtually existing molecules in a library, which were commercial vendors, and to see which one would fit best. This was done by docking and designing. Then we ordered the molecules and we tested the molecules. And I think that two things were happening. In the beginning, we gave the data to the modeler to see if he could improve his docking algorithms based on the data. You have then results, physical results, and you can feed them back into your program and optimize your program. But it was then This was initially, but then we got this crystallographer who could actually crystallize the compound into the pocket. And of course, then we can really see where the pocket is and which interactions, and that actually helped a lot. And it superseded then the docking. So we stopped docking once we had crystallography data.
Okay. Also, I've seen that the first compound that was synthesized, it did not have a carboxylic group. The first compound that was synthesized did not have a carboxylic group, but later, the next one that was gotten later had a carboxylic group. Did this increase the effectiveness of the drug?
Well, I know, but I showed you so that the compounds from the screen and virtual and testing had this. What was it? Had 3 nitrogens, I think, in a 5 ring or something. This is, I'm not a chemist, but this is a bioisoster. People tell me that this is has same chemical features as a carboxylic acid. But adding the carboxylic acid added a lot. I think it was responsible for having antiviral activity in the cell. So we did have in vitro activity of the compound, so inhibiting integrase LGF interaction, but to have anti-HIV effect, we needed to add the carboxylic acid.
Okay, thank you, sir. Finally, I would want to know the mechanism of the action of the drug on the viral molecules. What is the mechanism of inhibition?
I don't understand the question. The mechanism of inhibition of what?
How was the drug able to inhibit the viral load?
Which one? The legend?
Yes, yes.
Yeah. So legend has 3 mechanisms. It inhibits the interaction with LGF. As a result, you will have less integration, and also the integration will be on the wrong sides. Yeah? So this is 2 mechanisms. But it also interferes with the assembly, which is a late step. So it has 3 different mechanisms, and that makes it very potent.
Okay, sir. Thank you, sir.
You're welcome.
So there are not any more. Okay, there is one more.
Hello, sir. Can you hear me?
Yes.
Okay, my question is, instead of using integrate inhibitors to block viral genome replication or integration, why not to use the new technology or genetic editing technology CRISPR for blocking the viral integration? I think that would be easier, much easier than, for my understanding.
Okay, it's a good proposal, but it's not easier. As for the cure strategy, due to limited time, I only talked about block and lock and about shock and kill, but people are also doing gene therapy and CRISPR to try to cut out the virus from the reservoir. Yeah? So people are doing that. The problem, and why is it not used for the treatment? Well, the treatment is easy. We have drugs. It works. It only cannot cure. I think for the cure, the CRISPR is possible. The problem is delivery. I mean, we do not even know where all the virus is, where all the reservoir cells are. And so for the CRISPR, you have to deliver the CRISPR technology to the places where the virus is. So if you don't even know where the virus is, it's difficult. I mean, if you give a small molecule, it goes through the body. I mean, it depends on the pharmacokinetics and pharmacodynamics and whatever. But for a CRISPR, which you put in a vector or a nanoparticle, it's not that easy, and it's still being developed. But I think that in the future, for the cure, I mean, CRISPR can have a good chance that it will work. Yeah.
Okay, thank you so much.
Are there any more? Okay, thank you so much, Professor. This was really an informative lecture and a nice discussion. So have a good day.
Thank you. You're welcome. Bye.
Bye.
Have a nice time over there. Bye.